Hello, everyone. I think we have people trickling in to join us for this month's member talk. Hello, hello. As you're coming in, if you want to say hello in the chat bar and where you're watching from, please do. Hi, Louise. How are you today? Can you hear me?
Yes.
Great, great.
Doing good.
Okay, excellent. Okay, do we have audience? Let's see here. Oh, panelists and attendees. All right, so if you are just joining us, go ahead and say hi in the chat so that we know you're here. We're going to get started in just a moment. We're just letting people join us a little bit at a time. And okay. Hi, Anne. Thanks for being here. What do you think, Louise? Should we get started?
Let's do it.
Let's do it. Okay. Good morning, members. Thank you so much for joining us for this month's member talk. We are super excited today to be welcoming our curator of ichthyology, Dr. Luiz Rocha. Hi, Louise.
Hi, everybody.
Oh, good. We see everybody in the chat now. Great. And so today's exciting in part because Luiz takes amazing photographs. So his presentations are always like pretty delightful for the eyeballs. But also it's exciting because if you joined us last month, you heard our executive director, Dr. Scott Sampson, talk about and introduce our new 3 initiatives. And Hope for Reefs is one of those. And we are actually moving into phase 2 for Hope for Reefs because a lot of great work has already been done and you've probably heard about that in the past. But today we are going to take a deep dive, which I'm horrified to realize is a pun, into what's ahead for Hope for Reefs. And Louise, who is not only our ichthyology curator and a lead member of our Hope for Reefs team, but also a member of our really incredible deep dive mesophotic science diving team, is a great person to take us into that material. So I will turn it over to him in just a moment, but as a reminder, you can chat about anything you want and to each other and whatever you like in the chat function or the chat box. But if you have a question for Luiz specifically, go ahead and throw that in the Q&A and I will either work those in as he's talking, or if there are bigger questions that I think deserve more time, I'll save them for the end and we'll have some time after his presentation to get into that stuff as well. So with that, Luiz, thank you so much for being here today. And again, members, thank you and thank you for your support. And please take it away.
Thank you, Laurel. Thanks everybody for being here. It's super exciting. We're finally moving on to phase 2 of Hope for Reefs. Phase 1 kind of started winding down before the pandemic, and then we had to stop everything during the pandemic. So now we're glad to be gearing up and trying to really start the activities for to take phase 2 out of the ground, which it's going to be much bigger than phase 1. So it's super exciting. This is an initiative that I co-lead with Bart Shepard. He's the director of the Steinhardt Aquarium. And then all of our coral reef biologists at IBSS participate on it, all of our educators. We have a couple of exhibits about coral reefs. So it's like across the entire academy kind of initiative, and it's really good to be working on something like this, where the entire institution is focusing on the same problem and trying to handle it. So before I go into the meat of the initiative, I am going to, if my slide shifts here, there you go. I am going to give you a little background on coral reefs, what they are, where they are, talk a little bit about their biology, and then move into the actual initiative and what we're going to do to help reefs around the world. So coral reefs, they develop in tropical locations, which are invariably beautiful, not only the reefs themselves, but also the locations. They require certain environmental conditions to thrive, including warm water, but not so warm, I'm going to get there, and clear water. And the necessity for clear water is because they live in symbiosis. So they live in a really close relationship with algae. So coral, it looks like a rock. If you touch it, it feels like a rock, but it's an actual animal. It's a rock covered, the rock part of it is the skeleton, and it's covered with a thin layer of tissue that is an animal. And the animal has a really close relationship with an algae. The alga lives inside the tissue of the coral, and together they recycle energy, and then they form these fantastic structures that can be seen from space. The Great Barrier Reef, for example, can be seen from space. But because the algae, the zooxanthellae, it's the technical term for it, that lives inside the coral requires light to conduct photosynthesis to produce energy. So it's a sunlit battery, if you will. It requires that light goes through the water. So the light only goes through the water if the water is clear. So corals like clear water and they like warm water on the tropics. Because they like clear water and warm water, and we like clear water and warm water, almost invariably, coral reefs are in places where people are. And you know what happens when you put nature together with people? Bad things happen for a number of different reasons. So because of that, corals are declining worldwide for a number of different reasons. Because of overfishing, because of pollution, both plastic pollution, for example, and nutrient pollution. So corals, they live in almost a balancing act between corals and algae, not the one that live inside the tissue, but there's also other types of algae, of plants on the reef that kind of compete with the coral for space. And if you add nutrients, for example, pollution from the sewer to a reef, then the algae on the reef grow faster than the coral, and they can outgrow the coral. That's what that picture on the bottom right there is showing. So to have a good coral reef, you require low nutrient water. So waters with very little nutrient. And when we add sewer to the water, then we add nutrients to the water, the algae outcompete the coral and it destroys the reef. Physical destruction, those are all more or less kind of localized threats that are, I wouldn't say they're easy to deal with, but they're easier to deal with than the big threat that is global climate change. So on the left there, that's a picture I took in one of our last trips to French Polynesia. It was, I took that picture in April of 2019. And by all of what we could tell at that time when we went there, it was a healthy, thriving reef. You see, it's a beautiful reef, very colorful. All of the coral was alive. There was a lot of fish. You can't see a lot of fish in this picture because it's a wide angle view and the fish are small, so we can't quite see the fish very well. But there was a lot of fish in that reef. The diver there is Bart Shepard, the director of the aquarium. I used him for scale in my pictures. So we went there in April 2019 to study the reef scientifically, to collect specimens for the collection, to take a lot of data about the environment. And then we came back to San Francisco, and 5 weeks, 6 weeks later, we came back to the same reefs to collect fish for our exhibit. So we have a Twilight Zone exhibit here that hopefully you've seen. If you haven't, hopefully you come soon to see it. I'm inspired to see it, hopefully in the near future. We came back 5, 6 weeks later with a different mission. So we came back to the same location to collect live specimens for the Twilight Zone exhibit. And what we found was this on the right there. So every white coral you see on that picture is either dead or dying. And the reason for that is because the water got warmer than normal. So I said coral reefs, they like warm water, but they like stably warm water, and they don't like super warm water. So when the water goes 1 or 2 degrees above what they like, they get very stressed. The coral animal gets very stressed, and then they expel the algae that lives within their tissue. So the algae is actually what gives color to the coral. So when they expel the algae, you can see through the tissue of the coral animal and into its skeleton, which is white. So that's what's happening there. All of the corals, they expel the algae that live within them. And what's left behind is just the transparent coral tissue with the white skeleton behind. So we call that coral bleaching. And it's happening more and more, almost every year now. It's repeating itself, and it's damaging corals in an unprecedented way throughout the world. Some reefs declined by 70, 80% because of this phenomenon. It's something that's worrying scientists all over the world. I've been diving on reefs for the better part of 30 years, but I don't study the corals. My specialty is fish. So I never tried to see this happening because I knew it was going to hit me hard when I saw it. And this is the first time I actually saw it. It was just a couple of years ago. And it really hit. I got depressed on that trip for days after seeing this because it's something that's really sad. I mean, you know that this is extremely diverse, extremely beautiful ecosystem that is dying because of what we're doing to the planet. So this is basically what we're trying to reverse with Hope for Reefs, and or helping try to reverse. So the basic mission of the initiative is to provide sound science that leverages the power of community and global solutions to significantly reverse the decline of coral reefs. In a nutshell, we think that this is not going to be solved by like top-down government regulations. It has to be something that comes up from communities, and it has to be something that is based on science. That's the basic premise of Hope for Reefs, that we provide the best possible science to come up with the best possible solutions that are, for the most part, community-based. Because if they're not community-based, then they get disconnected from the communities and they're not enacted the way they're supposed to. So when I get to this point on the presentation, I talk about services that coral reefs provide to humans, like, I don't know how many thousands of miles of coastline they protect, or how many hundreds of millions of people they feed. But I'm not going to do that today. I'm going to do something that hopefully you remember. It's those boring facts people usually don't remember. So I'm going to tell you a different story. And for that, I wanted to take you in this virtual vacation, if you will, to this beautiful location in the tropics. It can be, I don't know, in French Polynesia, Bora Bora, or in the Maldives, or in the Bahamas. You just pick your dream location, or the best beach you've ever been to in the tropics. Remember the white sandy beaches. Maybe imagine yourself sitting in one of those chairs there, sipping your favorite drink, your favorite cocktail, your I don't know, fizzy water, whatever rocks your boat on those places. Maybe walking around that warm white sand, and then realize that all of that sand is actually parrotfish poop. White sands on the tropics invariably are parrotfish poop. And that's because parrotfish, they eat coral, either dead or alive. They digest the tissue, the organic matter that is attached to the coral. Or if it's a dead coral, they digest the alga that's attached to the coral. So they digest the organic portion of it. And they have really powerful jaws. They're called parrotfish because their teeth look like beaks from parrots. They're really hard. So they kind of pulverize the coral within their mouths. They digest all of the organic matter. And then what they poop in the end is just pulverized coral, which is the white sand that you find on the beach. So that poop, they washes up to the beach. And that's what forms the white sand beaches. That's why we don't have white sand beaches in San Francisco. It's because all of that sand actually comes from land. And when the sand is formed on land, it's formed from a different way, through a different process. And it's basically rocks that are pulverized into sand. So it's brownish, kind of ugly, if you ask me. But in the tropics, it's pure white because it's pulverized coral. Now, normally, a coral reef, the parrotfish are really good for coral reefs because they make coral grow faster. But that only happens when the coral reef is very healthy. So it's the same thing with sharks. Sharks eat a lot of fish. But if you overfish a coral reef, there's going to be fewer fish and fewer sharks, and that's not a healthy system anymore. It's the same with parrotfish and coral. When the coral reef is really healthy, there's a lot of parrotfish eating a lot of coral, and the coral grows super fast, so everything works in balance in the reef. The problem today is that we're killing both the parrotfish through overfishing and the corals through climate change or pollution or overfishing or like anchoring on top of the coral, you name it. So we're killing both the parrotfish and the coral. And the idea here is that we want to partner with the entire, whoever we can, actually, to try to make sure that those beaches are there for posterity, for as long as we can see in the future. Now we're doing that through several different avenues. We do that through biodiversity. The most basic way of doing it is biodiversity data collection. So the first and best step you need to do to kind of figure out what to protect on a reef and how to protect and how to reverse the decline of a reef is knowing exactly what lives on that reef. And we have a lot of specialists here at the Academy that study everything from the coral itself to the mollusks to nudibranchs gorgonians, to urchins, to fish, all the way to fish, which is what I do. When all of our experts are together on a reef, we learn a lot about that reef. We can do really complete biodiversity surveys, and we use all of that data to try to find what's the best solution that applies to each specific place. Now, what's really unique about the Academy is that we do that all the way down to what we call the twilight zone, which again reverses back to the exhibit we have in the public floor, and go see it if you haven't. So most, I'd say 99.5, 99.8% of everything we know about coral reefs comes from the top 100 feet. That's where almost all of the studies about coral reefs are done, because that's where the normal scuba diver is restricted to. So if you go to a dive shop today and you say, well, I want to be a scuba diver, they'll train you at the open circuit diving, which is the most basic level of training you can do. It allows you to dive to about 60 feet. And then the next level, it's advanced scuba diving. It allows you to dive to 150 feet. So that's the depth where most scientists are restricted to. If you want to go deeper than that, you have to do technical diving, which is what we're doing here in this picture. That's Meridius Bell, a dive safety officer on the background there. He's wearing this crazy gear that I will talk a little bit about in a minute. It's called the rebreather, and we're installing arms on a reef at about 100 meters depth that is in Palau. And these structures, so we deploy them on the reefs. We're deploying them at about 100 meters depth there. We leave them there for 3 years, so they become part of the reef. A lot of the organisms in the reef, they colonize it. Then 3 years later, we go back, we retrieve them, we take them back to the lab, and then we pick everything out of it that we can, and we identify everything we can by eye, and what we can't, we put it basically literally on a blender. We blend everything, and then we sequence the DNA of everything that ever lived on that piece of structure. So that gives us the best possible biodiversity survey you can think of, because it detects everything that lived on that particular area for the past 3 years. So this is going to give us like unprecedented information about what lives on not only on the shallow reefs, but also on the deeper reefs, going all the way down to 100 meters, which is something that we are one of the few institutions that do it. To give you an idea, right now, about here at the Academy, we have 8 or 9 divers trained to do this type of work. This is the largest, most active technical scientific diving team in the world right now. There's no other institution that has more scientists diving to those depths. There's no other institution that has scientists studying multiple different organisms, looking at their evolution, at their ecology at those depths. We are like #1 by far. So this is more or less what the twilight zone is. So that the top 100, maybe 200 feet, you were a little bit crazy with the open circuit scuba. It's everything we know about reefs. So it comes from that top 100 to 200 feet. Between 200 to 500 feet is technically the twilight zone, which is the technical term for that is mesophotic coral ecosystems. And then below 500 feet is like almost deep sea kind of work. And a lot of people ask me, well, why don't you use, instead of using all of that technical diving gear, why don't you use a submarine or a remotely operated vehicle to study those depths? For a number of different reasons. First of all, using a submarine is very expensive. It's upwards of $20,000, $30,000 a day, plus the cost of the ship. So it's very much more expensive than the diving. The diving that we do is more expensive than normal diving, but a lot cheaper than submarine. But more importantly to me, the organisms that I'm studying, the animals that I study that I'm interested on are fish. In fish, they swim, and they swim away from the submarine. The submarine is very noisy. It's big. It has lights. When you get close to a reef on a submarine, which I did, all of the fish hide, so you can't really study them. So if you wanted to study corals or gorgonias or sponges or animals that don't move, a submarine would be fine. But to study fish, it's really not the right tool for the job. It's like trying to study birds on a rainforest with a helicopter. Right? You can study the plants, but really not the birds. So it's the same thing with the submarine at those depths. And because it's so expensive, whenever scientists have access to those submarines, they'll take them all the way down to the deepest depths they can. So the end result of that is that we know a lot about the shallows. We know more or less about the deep, but we know nothing about this in-between region, the mesophotic coral ecosystems. So the technical diving portion of it is really hard because it requires a lot of gear. And back in the day, it used to be done with open circuit gear. And that's the gear that when you breathe in, the air comes out of the tank and goes from the regulator into your mouth. And then you exhale, it goes out in bubbles. But we have to spend a long time in the water decompressing. So we have to take all of these tanks, and then it's almost like diving with a submarine. You can't move around, you can't chase the fish. So you need to find a different way. So the solution we found was this equipment that's called a rebreather. It is much smaller, much more compact than trying to dive with all of those tanks. And it's quite a simple apparatus. So instead of the air coming out as bubbles like it does when you're diving in open circuit gear, when you exhale, that gas that you exhale goes back into the canister on your back. And if you want to see this actual equipment again, I mean, I don't know how many plugs I gave to the exhibit here. It's the 4th or 5th plug, but it's actually on the exhibit there. The funny story about the exhibit, that the mannequin that is on the exhibit with the actual gear, the exhibits team bought all brand new gear for the mannequin, and it's expensive gear. So I told them, oh, you want to really be original? You have to use used gear there. So I gave him all of my old used gear. The mannequin happened to be my size. I gave him all of my old used gear and then grabbed all of the new gear. And the gear that you see on the exhibit today is gear that I actually use down to those depths. And the used gear, the new gear, I'm using it. I'm not using it anymore. It's already trashed, but but I used it for quite a number of trips. So when you exhale the gas, so when you breathe normally, you inhale the gas that's in the atmosphere, which is 20% oxygen, 80% nitrogen, and then you metabolize that oxygen, and then you produce carbon dioxide, which is not good for you, and that's why you exhale it. So you don't want to be breathing the same carbon dioxide over and over again. So when you exhale it on a rebreather, there's a filter on our back that removes the carbon dioxide. And then there's electronic sensors that constantly monitor the amount of oxygen in the mixture, because they want to give you the correct amount of oxygen. So the oxygen sensors monitor the quantity of oxygen, and then they inject only what you need in terms of oxygen. So just a tiny bit of oxygen every now and then you keep breathing that pure, good mix of gas over and over again. And that lasts for 6, 7, 8 hours. The limitation is the filter. So with that system, we finally were able to go to those depths and study it. And one of the first things we wanted to test was if it was a refuge or not. So there was a lot of talk in the past that indicated everybody thought that those deeper reefs were a refuge for the shallow coral reef fauna. And that was based on basically 2 assumptions. One was that everybody thought that the deep fauna was very similar to the shallow fauna in a coral reef. So the deep fish were very similar to the shallow fish. And the other one was that those deep reefs are a refuge. And when we started doing this work, we saw that none of these 2 things actually happened. When we went down and we counted Luiz, the fish, we did all of that. We looked at what was actually happening there. We saw that none of these things actually happened.
And Luiz, can you hear me for a second? Yes, I was just going to jump in and ask you to define what we're saying a refuge. So a refuge from what in this case? Okay. So what that actually means.
Okay. Yeah. So because of all of those impacts that are damaging the coral reefs, we're seeing them in the shallow reefs, and nobody is diving to the deep reefs. Everybody thought that those deep reefs were protected. They were not as impacted as the shallow reefs. So everybody thought that there would be more fish in the deeper reefs, and that even if we destroyed the shallow reefs, the deeper reefs would be able to reseed the shallow reefs with the same animals. And then at some point, if we protected the shallow reefs, the deep reefs would revive the shallow reefs, because everything was shared between them.
Right. So they thought like maybe the same species that existed in the shallow could like hide in the deeper and be safe there to just repopulate whenever it got better. Right.
Exactly.
Okay. Thank you.
Yeah. Yeah, so one of the first things we found out was that it's not the same fish at all. To the point where all of these fish here that you see, it's like 12 of probably 15 or 16 fish that we described, we named. There were no scientific names for these fish. We were the very first to collect them from all over the world, several different places we've been to. Every time we go, we find new species. So those are just 12 examples of some of the most recent new species we found doing this work. And then when you look at the entire fauna, those numbers there show how different they are. So the shallow reef is 87 to 96% different from the depth between 30 and 60 meters. And then the deeper you go, the more different the reef becomes. So between 60 and 70 meters, it's 97 to 99% different than the shallow reef. And then the deeper you go, the more different it becomes because the environmental condition changes so much. The temperature gets lower, the water gets darker. So it's really a completely different fauna.
So what that actually means is that like a loss is actually a loss.
Yeah, a loss in the shallows is actually a loss. Yes, yes. They even if there were no impacts on the deeper reef, there wouldn't be that receding from the deep to the shallows because it's different set of animals adapted to live on that depth. So let me give you an analogy. Let's say that our goal here was to protect rainforests. And then everybody said, oh, we don't really need to protect them because the savannas are protected and they're a refuge for the rainforest. But if you look at it in detail, they're in more or less the same area. There's savannas and there's rainforests in Africa, but the species that live on them are very different.
Right.
And they're both under threat.
Right.
Which is the second point of the talk here. So are they really out of reach? So because everybody thought that this was what deep reefs look like, I mean, it's like nobody knows what's there. Everybody assumed that there were no impacts there. And then we actually went there and we saw it was not the case. One of the trips we did to the Bahamas, we did it right after a hurricane had passed. And hurricanes it's an event that we know that severely damages shallow reefs. Because of the waves they create, the currents, they break a lot of coral. So it's very damaging to the shallow reefs, but everybody thought they wouldn't impact the deep reefs at all because the waves don't reach the deeper reefs. So the waves don't reach them, that's true. But all of the steering that the hurricane does in the water on the shallow reefs, it steers up all of that sand rains into the deeper reef and smothers them. So this is not what a deep reef is supposed to look like. It's not supposed to be white like that. All of that white in that picture there is sand that kind of rained down from the shallow reef after the hurricane passed and then buried the deep reef. And when sand buries a coral like that, the coral invariably dies. So natural impacts get down to the deeper reefs that we thought that they wouldn't. A lot of the bleaching that we see in the shallow reefs is also affecting the deeper corals. A lot of the diseases that are affecting the shallow reefs are also affecting the deeper corals. Anchoring, this is a picture I took in Palau at about 110 meters depth. That's a rope from an anchor on the left side there. So the destruction that we see in the shallow extends to the deep because people are kind of reaching into the deeper reefs more and more these days. This is one of the deepest pictures I've ever taken. It's over 500 feet depth in the Philippines. That's a decorator sea urchin. So it's one of those sea urchins that kind of grabs everything around it to camouflage itself with. And that blue thing there is a piece of plastic and it's hanging onto a fishing line. So that's at about 150 meters depth. It's just sad. It's one of the saddest things to see. So I'm going to tell you a story about a trip that I did in 2017 to St. Paul's Rocks that kind of summarizes the whole thing. St. Paul's Rocks is a tiny island in the middle of the Atlantic, surrounded by water. It's about 1,000 kilometers from the Brazilian coast, 2,000 kilometers from the African coast. So that's what, 600, 700 miles from shore from Brazil, over 1,000 miles from Africa. So it's basically in the middle of nowhere. The only building in the island is this tiny research station for 4 people. If I turned around and took another photo behind me there, all you'd see would be ocean. So that's the whole island there. It's maybe the area of a football field. That's about it. And this is a video that we took during one of our dives. And I'll kind of let it go here. I should have the sound enabled, but you might need to make your volume higher a bit because you're going to hear some interesting things there. So this is us diving. I'm gonna pause it right here. That's the depth, 419 feet. The person that's filming is Mauritius, the one that was in the picture before. And then Hudson and I are diving and we're collecting fish, and we're in for a big surprise there. So Mauritius is filming. He gets close to me. That's me catching a fish. And then he sees this massive shark. And if you can hear it, the helium voice. So we have to breathe helium at those depths. And he's telling me, look at the shark, look at the shark. And I can't understand what he's saying. I'm thinking he's saying, let's go up. Because every time we talk to each other at those depths, it's usually, let's go up. So I thought, no, I'm okay. I don't want to go up. I'm catching a fish. And then he does the same thing to Hudson, and Hudson's catching the same fish. And I'm paused it right here again. So if you look closely, the shark, there's a line coming off of the corner of the mouth of the shark. That's a fishing line. So this species of shark had never been recorded in this island before. It's the first time we're seeing one of these there. And it has a fishing line attached to its mouth. It's a shark that lives in the tropics. It lives only in deep water. It's a 6-gill shark, so it likes cold water. And in the tropics, cold water is deep. So we never see it in the tropics. And it's already with a fishing line in its mouth. It just kind of comes around. You can see the fishing line attached to it. So it's a shark that we hadn't seen before. Nobody had ever seen before in that depth. Really unique. Neither Hudson nor I saw the shark on that dive because we were so distracted by the fish. We only saw it on video after. This is the reason why we didn't see the shark. We were catching this fish. So it's one of the most, I think it's the most beautiful fish I have ever named. And I've named quite a few. I've described scientifically quite a few. We named it Tosonoidis Aphrodite because it hypnotized us like the Greek goddess would. And when we ignored that giant, massive shark. So untouched, not really. I mean, fishing line on the mouth of the shark. And then same, not really. It's an entirely different set of species. So going back to the initiative, and then back to that community focus, in the Philippines especially, and then expanding that to other locations now, we do a lot of that community effort. Mostly because in the Philippines, the management, the fisheries management is done at the community level. So we've worked really closely with communities there. And whenever we show them our discoveries, most of the time when it comes from a shallow reef, they know what it is. It's not like we're discovering something entirely new. Even if it's new to science, it's not new to them. But when it's from the deeper reefs, it's actually new to them because nobody ever goes there. But they get really proud of it, and they get this sense of ownership. This town here in the Philippines is Puerto Galera, and when you get to it, there's no airport there. The only way to get to it is by boat. When you get there, there's a sign saying, "Welcome to the center of marine biodiversity in the world." And this is actually the place where we found the most species anywhere in the world in a coral reef. It's the center of diversity, and they're proud of that. They created a lot of marine protected areas around, all through community effort that mostly Terry Gosliner, one of our coral reef specialists that specializes in nudibranchs here, has been doing that for the last 20 or 30 years or so, which even resulted in our Philippine coral reef exhibit. So if you didn't know, the big tank in our Philippine coral reef exhibit, it's built, the matrix of the tank, all of the concrete that formed that structure in it, is built actually based on photos that we took of an actual reef in the Philippines. All of the fish in there, with very few exceptions that got there by accident, but all of the fish in that, almost all of the fish in that tank actually come from the Philippines. So it's entirely based on the Philippines. Other activities we're doing as part of Hope for Reefs that we plan to expand going into phase 2 are the coral spawning lab. So behind the scenes in the academy, we have a lab where we artificially induce coral to spawn. It's like amazing because corals, they get cues from the environment to spawn. So it has to be the right light. It has to be the right phase of the moon. So we have lights that mimic the phase of the moon, and we change the temperature according to the season of the year that they're supposed to be. When it's winter, we kind of drop the temperature a little bit. Summer goes higher. Full moon, we put more light. New moon, we put less light. So whenever it gets to, these corals are from Palau, whenever it gets to May, which is the time when they spawn in Palau, they spawn here in the lab. And then we experiment with those eggs, trying to figure out what we can do to help them survive the increasing temperature of the water. We work in partnership with several different institutions, including Secor, Autodesk, that made those like 3D printed artificial settlement plates to put corals on. So there's several ways you can restore a coral reef. One of them is just by fragmenting. So if you remember some of the pictures I shared, the coral reefs, some of the coral animals, they're really branchy. They have a lot of branches. So if you break one of those branches and you plant it somewhere else on the reef, it's going to grow into a different coral animal. But when you do that, you're just creating a clone. So it's a piece of that coral that's going to result into a different coral individual, but it's going to have the exact same genetics as the other one. So with what Secor does is it grabs those eggs from the coral and then plants them on those little structures. And then we plant those structures back into the reef at a large scale. And the structures, they have to have those shapes, because if you put something that is just like a plate, if the plate goes upside down because of a wave, then the coral that was on top of it is going to die. And those structures, they're self-stabilizing. So even if we throw them on a boat, they stick to the reef and then they don't roll over. So we can do that at a large scale. So we can plant 1,000 or 2,000 corals at a time instead of just one at a time. And then we work with several other institutions trying to develop this technique to better restore coral reefs. Now, with all of the bad news that we hear about coral reefs, including that picture that I showed you with all the bleaching coral, a lot of people ask me, I mean, is there even reason to have hope for reefs? And to convince you that, yes, there is hope for reefs, I'm going to tell you another story. I like stories, if you haven't noticed. I'm going to tell you another story here, and that is the story of Bikini Atoll. So this is the largest man-made nuclear explosion ever detonated on the planet. This is Castle Bravo. It was an explosion, a test bomb created by the US military in 1957. It was 1,000 times more powerful than Hiroshima. Massive, massive explosion over Bikini Atoll. It left this huge hole in the reef there. It's Bravo crater, over a mile wide. The red circle there is the actual crater. It's about like 200 feet depth. And the yellow arrow on the outside there points to a place where I dove just 49 years after the explosion. And when I dove there, I found something that I was not expecting. So this is what I found. Remember when I said sharks on a reef are a sign of a healthy reef? This was one of the healthiest reefs I've ever seen in my life, in Bikini Atoll, less than half a mile from the site of the biggest explosion there ever was on planet Earth, caused by humans. The explosion was so powerful that the pulverized coral that resulted from it rained on and affected the population of islands that were about 300 miles away. That's how powerful that thing was. And people had to move out of not only Bikini itself, which there was people there amazingly, and people didn't know really what the effects of radiation were back then, but they had to move out from islands that were 300 miles away from the actual explosion site. So I did not expect to see this. I expected to see like a coral reef, but not a thriving coral reef. And this is what I saw. And this is one of the things that gives me hope for reefs, is that the only thing we did there in Bikini was leave the reef alone for 49 years. And when we did, it almost 100% came back. So if we find the right intervention for the right place, using the best possible science, the result is hope for reefs. And this is, on a nutshell, what we want to do, is to find the right intervention for the right place using the right science. For Bikini Atoll, the right intervention was close it, take everybody out of it, stop fishing for 49 years, and it came back to 100% normal. For the Philippines, it might be better fisheries management. For the Caribbean or Curacao, it might be we need to make coral restoration because there's so few coral left. What we really need to do is the most cost-effective way and the best way to do it for the best location with the help of the community. So I don't know, maybe Scott talked about the new strategy, right? With the new, the three pillars of the strategy, the three kind of ways of conducting work at the Academy, three ways that focused on biodiversity science, environmental learning, and collaborative engagement. So Hope for Reefs and all of our initiatives, they kind of followed along those three pathways. And I'm not going to read that. We're probably going to share this with you in written material at some point. And then the ultimate impacts we want to Hope for Reefs, the initiative, have is that worldwide ecological health of coral reefs is kind of shifting from declining right now to regenerating, to regrowing in the future. We really hope that human communities, they start embracing diversity, coral reef diversity and human diversity as a path towards a regenerative future. And we really hope to establish a new, deeply collaborative, community-based conservation strategy that's being widely implemented in coral reefs around the world. And with that, I will take questions.
Thank you. That was very inspiring. And yeah, so we do have two really good questions from Mary, who wins the question award at this point. But I just want to say, tell everybody, like, this is a really, there are no dumb questions. Like, we know the initiatives themselves are very large and very sprawling, and they're really cool and really inspiring. But if you have questions about any level of them, whether it's the actual science, whether it's questions for Louise about diving, whether it's, you know, how-tos or in country, out of country, whatever, like, feel free to just ask them right now, because this is a great opportunity to just have a really informal kind of Q&A with an expert. So I will start with, Mary's first question was about the arms. And she asked, is it known whether there are certain corals or organisms that will not colonize an arm structure?
That's a good question. Probably not, because invariably, so the arms, it's not a technology we developed, it's a technology that was developed by the Smithsonian, and they are using it in hundreds of locations worldwide, but they're only doing it in shallow reefs. And whenever they do it in shallow reefs, invariably, it captures the diversity of shallow reefs like over 100%. And by over 100%, I mean that the estimate of diversity you get from the arms is always better than what we had before. That was the accumulation of knowledge over the past, I don't know, 10, 20, 50, 100 years, depending on which coral reef you are. That's how good they are. And the reason for that is because we don't only look at the organisms that attach to it, we look at the DNA. And the technique is so sensitive that even if it was a coral that kind of settled there as a baby, but didn't grow because it didn't like the structure, it still leaves the DNA signature behind, and we can read it and we can detect it. So because we can detect everything through DNA, if it was a fish that swam by and the scale of the fish dropped into the arm and then it kind of got capturing there, we capture the DNA. That's so incredible. Because it's so sensitive like that, we have like this really good estimate of what lived in the whole area for the past 3 years.
That's so remarkable. Did you ever think, when you started as a scientist, as a baby scientist, did you ever think one day you would just blend things and sequence everything?
No, not incredible. By a long shot, not by a long shot. And this was unthinkable even 20 years ago. So just to give you an idea of how much this the genomics field evolved in the past 20 years. The first human genome, the original human genome, the first one to be entirely sequenced was sequenced in 2002. And that cost $2 billion with a B. And today you can sequence a whole entire human genome for less than $1,000.
Yeah.
That's how much the technology evolved in 20 years. And of course, it was all developed for humans, but we're glad to use it. I mean, every animal out there has DNA, so it's the same technology.
Absolutely. Thank you. And then I want to ask Mary's second question, which is great and specific to the coral spawning lab. She asked, are the temporal mimicking procedures done in real time, and can those be sped up?
It is done in real time. We haven't tried speeding up yet. What we, and it would be great to speed up because it would have multiple spawns during the year. So we haven't tried speeding up yet. We don't know if it's going to work. But what we do is to have multiple spawns a year is to get corals from different locations. So if we get the corals from Palau, which is in the northern hemisphere, they spawn in May. But if we get the corals from Australia, which is in the southern hemisphere, they spawn in August. So then all of a sudden now we have some tanks with coral spawning in May and some tanks with coral spawning in August. Then we have multiple spawns during the year and we can study them throughout the year like that.
Right. Yeah. I should mention too, we actually have a live cam on the coral spawning lab when it's expected to spawn. And although this year the pandemic really interrupted being able to do the work and physically, because it requires people to physically come in and all kinds of other shipping things. But we have some recordings of past ones that are really amazing, and we will be sure to, you can always watch our social accounts, and we always message when those coral spawning windows begin. So keep an eye out for that.
Yeah, the Academy scientist that's responsible for that is Rebecca Albright. And I think she has a talk about it, right? Recorded somewhere. So if you're more curious about that, you can go back to Rebecca's talk. And
yes, we did a breakfast club on it. I'll try to drop that in the links later. Okay, here's a question for you from Josie. How many species of corals are there?
There are about over 400 species of coral. But they're very unevenly distributed. So I said the Philippines is the one with the highest diversity. So in the Philippines, there's about 350, 400 species. And in the Caribbean, there's only about 40 or 50. And in Brazil, there's only about 15, 20. So they're very unevenly distributed. And the highest diversity is in the Philippines and Indonesia.
And I don't know that you mentioned this in this presentation, but one thing that is always so amazing is the kind of average rate of discovery when you dive to those mesophotic depths, because it is all so new. Can you remind us what that is again?
Yeah. So we have a chart that we show sometimes, and we show how many hours we dive or how much time we spent at the actual depth. So when we do those deep dives, I say that the dives are like 4 hours, 5 hours long, but we don't spend all of that time at the bottom. We spend only 5 to 10 minutes at a time at the bottom, and then all of the rest of the dive is decompressing. So we get so much gas diffused into our body under pressure that we can't come up fast. If we come up fast, it would be the equivalent to getting a soda bottle, shaking it, and then open it and just explode. So that's what happens to our blood if we go down and come up fast. So we have to go up really slowly. So imagine opening the soda bottle just a little bit at a time after you shake it. So we have to come up really slowly and stopping every 10 feet. And the computer tells us how many stops we have to do and for how long, to make sure that none of that gas goes back into out of solution as bubbles. So we don't want bubbles circulating in our bloodstream. That is not good. So to avoid that, we have to go up really slowly. So it's 5 minutes, 10 minutes at the bottom at a time, and we still invariably catch new species. So the number that Rich Pyle came up with, which is one of the pioneers in this field, is the number of species collected per hour of diving, number of new species collected per hour of diving. And it ranges from 4 to 5 new species per hour to all the way to like 15 new species per hour of diving. It's just incredible. And that's for fish. So if we started collecting, fish is like the birds, right? If we started collecting crabs and shrimp and like all of these tiny little things, which we will collect with the arms, then the number would be hundreds of new species per hour of diving. Right.
And your what you said about decompression is a nice segue into Elizabeth's question. How do you protect the fish from getting the burns when you bring them up from hundreds of feet below?
Right. So when we bring them for scientific purposes, we don't need them to be alive, so we just put them in a bag and bring them up. But if we want to bring them back to the exhibit, which we do a lot, I think over half of the fish that we have in the exhibit, in the Twilight Zone exhibit, they're not exhibited anywhere else in the world. You can't go to a dive shop, to an aquarium shop and get one, because none of the aquarium collectors are going to that depth to collect them. So we're the only ones that collect them. And then to bring them back alive, we put them in a fish decompression chamber. I don't think I have one in my office. I do have an arms thing though. I'm gonna bring it. I like props. Okay. We have a decompression chamber for the fish. This is just to give you an idea of the size of the arm thing. So this is an assembled arm. It's super heavy. It's like 30 pounds. It's a pain to put it down in the reef, but I don't have a decompression chamber here. I think Bart has a decompression chamber in his office, but there is one in the exhibit, another plug for the exhibit. And then we slowly decompress them over days. So because we spent 5 minutes under, we have to decompress for 5 hours. The fish spent their whole life under, so they have to decompress for a few days. And then we do that in the chamber. So we seal them inside the chamber. We hook up pumps to it to keep the pressure high inside the chamber. And then we kind of circulate the water and feed them through the pumps and everything. And then we keep them in the chamber until they decompress. So very slowly, like 10 feet per day, ascent rate or something like that.
Yeah. I want to ask this really good question from Anne. How does coral reef health relate to other tropical ecosystems like mangroves or seagrass?
They're highly linked. So both mangroves and seagrasses are a place where there's a lot of baby coral reef fish. So the baby coral reef fish, the coral reefs are a more dense ecosystem. So there's more sharks, there's more big fish, there's more groupers, there's more predators in general. So the baby fish are safer when they're in a seagrass or a mangrove. So a lot of baby coral reef fish, they start growing in a seagrass and a mangrove, and then they move on to the coral reef later. There's a lot of, I talked about how a lot of nutrients is bad for the coral reef, but a little bit of nutrients is good for the coral reef. So the mangroves and seagrasses, they produce nutrients, and they produce nutrients at a rate that's really good for coral reef. So whenever a coral reef is close to a mangrove, there's a lot of nutrient input from the mangrove that makes the coral reef grow faster, and not enough for making the algae outcompete the coral. So they're tightly linked, and all three of them are actually under severe threat.
Yeah. Jim asks, where are you seeding with the new sea core structures, and have you had success?
We haven't tried them yet in the field. So we have developed those. We know that they're self-stabilizing, but we haven't, I don't think we've tried them in the field yet. That's probably a better question for Bart.
Okay,
but,
right, because it's pretty new technology, or
it's pretty new technology. We know
right?
that they all self-stabilize if we throw them, so they all have the right shape, but we don't know if there's any preference from the coral. The baby corals are very finicky. So in their larvae, they swim around and they try to find the best surface to settle on, and we don't know if they're going to have a preference for one of those different shapes or not. And that's why we need the coral spawning lab here, because we can expose the larva when we produce them in the tank. We can expose the larva to the different shapes and then see if they prefer one or another, and then use the one they prefer. But we haven't done those tests yet.
Yeah, and a lot of that prototyping and the work with Autodesk, was that Rebecca's postdoc, Dr. Taryn, can you remind me?
Taryn, yes. Forgot her last name too, but it was Taryn. Yeah, and Rebecca has a new postdoc now that is looking at differential survival. So she's trying to see if in adaptation, but not like evolutionary adaptation through the lifetime. So she's basically trying to see that if they expose the larvae to higher temperatures, if they're going to be immune to higher temperatures when they become corals, which would be really good. So you could like induce them to spawn in the lab, then expose them to high temperatures, and then replant them in the reef as heat-resistant corals. So that's one of the experiments she's doing.
Neat. Okay, I'm going to put her full name mention her full name in a moment. Charles had an interesting question too. Do we have any plans for cold water reefs, or where do cold water reefs fit in?
That's a good idea. If you've seen a lot of my talks, you know that I hate cold water. I'm supposed to go diving in Monterey tomorrow. Oh my goodness. No, I mean.
Oh no.
Yeah, exactly. It's so cold. It is a super interesting question. In general, the colder water corals are suffering less from this problem, mostly because most of them are not what we call zooxanthellate coral. So they don't have the algae inside their tissue. They have the skeleton. They grow the same way that the tropical corals do, but they grow a lot slower, and they don't have the symbiotic relationship with the algae. So even though waters are warming, even in cold water temperatures, they're not causing this breakdown into the symbiotic relationship, and not causing them to bleach, and not causing us to lose cold water corals at the same speed as we are losing tropical ones. So the big problem really is tropical corals.
Okay, okay, great. And so that was Dr. Taryn Foster, who's
Yeah.
actually a friend of mine. So I should have, that was a bad failing. But really, really interesting work, and you'll hear more about that for sure. I want to ask, let's see here. Simon had a question. Is there any risk of accidentally populating a reef with coral polyps from other oceans during seeding projects?
That is a really interesting problem. I mean, it's a really interesting question. There's no risk, but there's people that actually want to do that. Which I'm not in favor of for a number of different reasons. I think when we try to do things like that, we always end up shooting ourselves in the face. So when we try to, for example, control, like there's a famous case in an island in Brazil that we went to recently, Fernando de Noronha, that the island had an infestation of rats. The rats are not native to the island, but they were introduced when people came in on boats. So there was a lot of rats in the island. So they brought a lizard from the mainland, a big lizard that we used to eat rats in the mainland. So when they introduced the lizard to the island as a solution to eat the rats, the lizards found a much easier prey, which was the eggs of the seabirds.
Yeah.
So they become a second problem. So that every time this happens in Australia, there's several cases of rabbits being introduced, and then they introduce the dogs to control the rabbits, and then the dogs explode and start eating something else. So whenever we try to solve things with introductions, which is what people are trying to do here, there's talk. Some people are in favor of, for example, bringing corals from the Pacific into the Caribbean. And the reason for that is because a lot of corals in the Pacific, they grow faster than the Caribbean ones do. So they think that they would restore the ecosystems in the Caribbean faster. But I don't think so. I think that if we don't remove the threat, it doesn't matter which species you introduce. If we just keep doing the same thing we're doing, they're going to be doomed anyways.
And in some cases, are they trying to test species that are hardier or more
right
warm temperature tolerant?
Right. So to actually answer the question, whenever we do the reproduction and then the reseeding of the reef, we only do it on the island. So we don't bring corals from Palau to here and then back to Palau. If we bring anything to here, we only do experiments in the lab and we don't try to reseed those. So we develop the method here, but then when we do the actual reseeding on the ocean, we only do it there. So we kind of create a new lab on the actual place in many ways to avoid that kind of thing. Because when we bring them here, maybe there's a different bacteria that's going to grow on them that got into the water somehow, and then we bring that back to Palau, it might cause problems. So we never do that for biosafety reasons.
Right. Okay. This is really, really amazing. And I just want to take a second to point out that, like, members, your membership actually is a huge part of what supports work like this.
Yeah,
so
absolutely.
As you're hearing about all this and as you're hearing about the work and the hope and the advancements, like, that's yours too. And we're so grateful for the part that you play in that. I want to end with one big question from, well, this is from our membership director, Mindy, who seeded this question, but it's a really good one to end on. And it's, what can the average citizen do to help and support and protect coral reefs? Right.
Yeah, well, number one, you can vote. You can vote the environmental way. I hate talking about politics, but if you want big change, that's what you have to do. You have to vote the right policies, and you have to talk to your representatives and say that climate change is actually a problem and it's caused by humans. And if we caused it, we can revert it. So voting is probably the number one thing you can do. You can do small changes like going a less polluting car. You can do take steps to mitigate your carbon impact. You can reduce your emissions in many different ways. But the big changes are going to come when we have policy level changes. So definitely, yeah, do the small steps, like try to consume as little plastic as possible. I know it's impossible. When you go to a supermarket, everything is wrapped in plastic, even if you don't put them in a plastic bag. But yeah, I mean, try to reduce your plastic consumption, because it does. I mean, we know that plastic that goes in the water here at some point is going to end up in a coral reef somewhere. That's how connected the oceans are. I mean, World Ocean Day was yesterday, and they changed it. They changed the hashtag from World Oceans Day to World Ocean Day to really highlight the fact that it's one ocean and everything is connected. So the impact, the pollution, the trash that we throw in here, it's going to end up somewhere else at some point. It might be a coral reef or not, but everything you do in your life has an impact, and the more you minimize that, the better. And the number one thing you can do is voting and bugging your representatives to enact policy that is environmentally friendly.
And is informed by science.
Yeah.
Thank you. Yes, members are loving this, and Anne asked if this webinar will be available online, and we do upload all of these member talks to a special playlist just for members, so we can help circulate that to you, and absolutely. And Mindy has just weighed in and promised to bring Louise and the rest of the team back for future programs. So thank you so much again, Louise. Thank you so much, members, and we look forward to bringing you more amazing content about what the Academy is working toward and for very soon. Thank you.
Thank you.
Bye-bye.